Hi, everybody. My name is Ed Davis. I'm Senior Application Scientist at GeneCopoeia Incorporated, located in Rockville, Maryland, just outside of Washington, D.C. I want to thank you all for joining us for today's webinar entitled Advanced Tools for COVID-19 Coronavirus Research from GeneCopoeia. Before we get started with the webinar, I just want to go over a few practical matters. First, you may ask me questions at any time, but in order to do so, you will not be able to speak vocally to me. So instead, you will need to send me your questions in the chat box or the questions box in the Q&A button at the top of in your control panel. And secondly, even though you may ask questions at any time, in the interest of having the talk go smoothly, I will wait until the end of the talk, and then I will answer each question in turn in the order it's received. And when I do that, I'll read each question out loud, as well as provide the answer out loud so that everyone can hear. So with that, I just want to go ahead and go into today's webinar. Okay, so GeneCopoeia, if you haven't heard of us, we are a biotechnology company, like I said, located in Maryland in the United States. We focus primarily on DNA-based, nucleic acid-based tools for research in many areas of molecular biology and genetics, such as ORF clones, clones expressing microRNAs, microRNA 3' UTR targets, promoters, etc. And we have CRISPR tools as well as lentivirus production and many other services and products. But today, again, I'm going to focus primarily on everybody's favorite pandemic, the SARS-CoV-2 related COVID-19 pandemic, and the tools that we have developed here for research in this area. So I'm going to talk about the following main areas that you see on the screen. The first product area I'm going to talk about is called spike protein pseudotyped lentivirus. I'll explain what that is when I get to it. Then I'm also going to discuss coronavirus protein antigen microarrays. Next, I'm going to go into some diagnostic, qPCR-based diagnostic testing kits that we have for the COVID-19 coronavirus. And then finally, I'm going to talk about the COVID-19 related plasmids, which include many different product types that I just mentioned, as well as COVID-19 related lentivirus and adeno-associated virus. Okay, so we have a web page on our website dedicated to COVID-19 research tools. This is part of what it looks like, at least above the top half of the page, and it has different product categories. And so you can access some useful information as far as the general categories, as well as links to webinars such as this one, as well as FAQs. Okay, so let's do a little bit of background about the SARS-CoV-2 virus, which is the central player in all of this. So this, of course, this graphic, we've all seen this literally everywhere on the news, in publications, advertisements, etc. It comes from the U.S. Centers for Disease Control, depicting the SARS-CoV-2 virus, which is a classic coronavirus member of the genus beta coronavirus. It's a spherical structure with these spike-like proteins. In fact, they're called spike proteins surrounding the surface. These proteins engage with the receptors on cells. And the SARS-CoV-2 virus is highly similar to SARS-CoV-1, which caused the 2002 to 2004 SARS outbreak. SARS stands for Severe Acute Respiratory Syndrome outbreak. And in fact, they use the same cellular receptor, which is known as ACE2. It's a little bit less similar to another coronavirus called MERS-CoV. And this caused the ongoing MERS outbreak, which stands for Middle Eastern Respiratory Syndrome outbreak that began in 2012. So of course, as we know, these are much smaller outbreaks. The SARS outbreak, while it was devastating, it caused a total of about 8,000 known cases. And the MERS outbreak is about 2,500 known cases so far worldwide. So again, as I mentioned, the virus is surrounded by these surface glycoproteins known as spike, which is essential for virus entry, and it has a 30 kb genome that consists of positive strand RNA. So this is a schematic representation of the SARS-CoV-2 viral genome. Again, there's a number of known ORFs as well as that are produced as either polyproteins or discrete entities. The PP1AB is a very large ORF that's post-translationally processed into several ORFs known as these non-structural proteins, NSP1, et cetera, up to NSP12. And then there's the spike ORF, and then as well as the envelope protein and the matrix protein and the nucleotide capsid. So this is the basic genomic layout of the SARS-CoV-2 virus genome. Now, so here's a little bit about what's known about the interaction of the virus with the host. As I mentioned, the SARS-CoV-2 virus spike protein has been shown to bind to a protein known as ACE2, which stands for angiotensin converting enzyme 2. It's the same receptor, as I mentioned, as the SARS-CoV-1 virus, but it's different than the receptor for the MERS-CoV virus, which uses a receptor known as DPP4. Entry into the host occurs via lung airway epithelial cells, although we also know that ACE2 is highly expressed on other tissues such as vascular epithelial cells and kidney, or as well as colon tissue and other tissues, which could possibly explain some of the severe secondary pathologies that we've seen other than respiratory failure, such as blood clotting and kidney failure. And that could be one reason why people with comorbidities are more susceptible to mortality from this virus infection than others. Another important protein for viral entry into cells is known as tmprss2. It's a serine protease that's required for priming of the binding to the spike protein to ACE2. And this is shown here in this simple schematic from Paper and Cell that appeared a couple of months ago. So the first product type I'm going to talk about now that we're through the background, is spike protein pseudotyped lentivirus. So what is this? So basically, this is lentivirus that expresses the SARS-CoV-2 spike protein in its envelope. Okay. So normally, lentiviruses, you may know, are highly derived from HIV. Okay. But lentiviruses can be engineered, and I'll go into this in a little bit, to where you can change the membrane protein that binds to cellular receptors to other viral entry proteins. And the SARS-CoV-2 spike protein is no exception. So you can change the tropism of the cellular tropism of lentivirus by changing the membrane protein to SARS-CoV-2 spike. So what is this useful for? Well, it's useful for several applications in a safe environment. So lentivirus is considered safe because even though it can infect cells in your body, it's engineered so that it cannot cause new viral replication once it infects cells. So normally, working with live COVID-19 virus requires biosafety level 3 conditions, and as we know, it's a potentially very dangerous virus. Well, if you want to study certain applications like vaccine development or the efficacy or mechanism of neutralizing antibodies against SARS-CoV-2, you can do this using lentivirus in which the membrane protein has been replaced with the spike protein. And it's also useful for development of antiviral therapeutic agents, as well as studying the mechanism of virus receptor interaction. So this is how it works. So if we look at lentivirus technology in general, again, lentivirus is a class of retroviruses that includes HIV. It contains a single-stranded RNA genome of 9.7 kb, and once lentiviruses enter cells, it integrates into genomic DNA, and so it can usually infect both dividing and non-dividing cells. Now, most typically used lentivirus is that most people use is pseudotyped by using the envelope from vesicular stomatitis virus, known as VSVG, that replaces the GP160 protein of HIV. And so in a plasmid system like this, where each component, different components of the viral packaging system are broken up into multiple plasmids, if you include this, if you change this VSVG to the spike protein, you can then package a transfer plasmid such as this one down here at the bottom that can contain a transgene, which can be anything like an ORF or a reporter like luciferase or GFP, into a lentiviral envelope. And so when we created pseudotyped lentivirus, spike pseudotyped lentivirus, we started out with a transfer vector that includes this vector known as P receiver LV201. So this expresses firefly luciferase from the CMV promoter and also contains a GFP. So what we can see then is we can use this virus to infect ACE2 expressing cells that we engineered and then look for both GFP expression as well as luciferase activity. And that's shown here. So if we have cells, so if we take the spike pseudotyped lentivirus, we then try to infect either normal, unmodified HEC293 T cells or such cells in which we've engineered them to express ACE2. We can then do a firefly luciferase assay on both transductions. And what we see is that the luciferase units measured for the unmodified cell line are in the background level, but we can get approximately 3 and even up to 5 times 10 to the 5 relative fluorescent units per mil from the spike pseudotype virus infecting the engineered HEC293 T cells. And that's shown here in the top panel. We have 3 different cell lines. This first cell line in panel A is this pseudovirus that's expressing luciferase and GFP that we use to infect cells that are expressing both ACE2 and tmprss2. Panel B is using the same virus to infect cells that express just ACE2. So we do get some infection. We can see some GFP expressing cells, but it's significantly lower frequency than the dual expressing ACE2 tmprss2 cell line. And panel C is a negative control line which does not express either ACE2 or tmprss2. And when we do luciferase activity shown down here in panel D, we see that the cell line that expresses both, that expresses just ACE2 gives about 20,000 relative fluorescent units, but we get about a 5 to 6-fold increase in RFU activity when we express both, the cells are expressing both ACE2 and tmprss2. And these two are negative controls that are not expressing either protein. Okay, so with this, we have created pre-made lentiviral particles that are available for next day shipping. You can buy them today, and if you're in the United States, you can get them tomorrow or the next day at the latest. And these contain several advantages. First, we provide accurate functional titers. So a lot of companies sell when they sell pre-made lentivirus, they just measure the copy number per mil using qPCR and then use that to extrapolate to a functional titer of transduction units per mil. That usually works pretty well, but we went a step further with this virus, and we actually measure the transduction units by FACS. So we do FACS sorting on the ACE2 expressing HEC293 T cells that are infected with the GFP expressing particles. So that way we get a very accurate titer of the virus. We also provide a complete system. So this can be used with GeneCopoeia's ACE2 expressing HEC293 T cells, which we also have available for next day shipping. And we also have kits for do-it-yourself packaging if you like to do it yourself. And it provides application versatility. So this can be packaged in either in vitro grade for doing, say, virus neutralization studies on cell lines, or we can even produce it in an ultra purified form that's suitable for in vivo grade. So if you want to infect these pseudoviruses into, say, mice and then do virus neutralization assays that way. And you can also express other commonly used markers such as GFP and luciferase, or most human, mouse, and rat ORFs in GeneCopoeia's more than 80 lentiviral transfer vector types. So we have a number of different versions of this spike protein pseudotyped lentivirus. So we originally produced virus in two different forms, either using spike protein in either its full length or a truncated version in which the C-terminal 19 amino acid endoplasmic reticulum retention signal was removed. Okay, and the reason we did that is we and others have found that the truncated spike protein actually gives higher titers, higher packaging efficiency. And these original versions used a spike variant known as D614. And D614 is significant because in original isolates of the virus from patients, amino acid 614 of the spike protein was an aspartate, or D, okay? But starting at about February, a new variant occurred known as G614, in which a glycine substituted for this aspartate at position 614. And that has become the dominant strain in not just in the United States, but in the entire world. So there are very few isolates now of the original D614 variant. And so it's hypothesized that this change, this mutation from D to G at 614 caused an increase in the infectivity of the virus. And in fact, in some publications, it has been shown that this D614 gene mutant variant does give higher infectivity in cell culture experiments. So we provide both D614 and G614 to customers, as well as the full and truncated. So we have 4 different versions of the spike pseudotyped lentivirus. Okay, so the next topic I want to talk about is the COVID-19 antigen microarray. Okay, so what are antigen microarrays? Well, we actually have a whole series of product lines on antigen microarrays, protein microarrays for various disorders like autoimmunity and allergies. Okay, so antigen arrays are a type of protein microarray. So proteins of interest are spotted on the membranes, and in this case, the proteins are known to be associated with specific diseases. And going even further, the COVID-19 antigen microarray contains a number of SARS-CoV-2 related proteins, okay? Antigen arrays are used for the detection of antibodies in patient fluids. So this is similar to a serology test, okay? So this would not be useful for looking for active virus, but it would be useful for looking for people who have had previous infections or who have had maybe other coronaviruses, such as seasonal coronaviruses. And they can also be used to validate specific antibodies. And usually the antigens are proteins that are known autoantigens in autoimmune diseases, but of course, as I'm talking about today, it can be other proteins associated with other disorders such as cancers or allergies or COVID-19. So the COVID-19 coronavirus antigen microarray contains 16 proteins spotted on the nitrocellulose filters. Okay, and several of these are SARS-CoV-2 proteins, such as spike protein S1, which is a highly variable region of the spike protein, as well as the receptor binding domain. This is the domain of the spike protein that interacts with ACE2, et cetera, et cetera. It also contains spike protein from SARS-CoV-1, as well as the nucleocapsid protein, and others such as MERS-CoV, and also the 4 known seasonal coronaviruses. And this is important because there is some cross-reactivity and antibody binding between the seasonal coronaviruses as well as the SARS-CoV-2. And also, to make this an even more powerful array, we've included antigens from influenza A and B, as well as RSV glycoprotein G. So this is available either as an array that you can purchase. The arrays come on glass slides, where we place 16 identical arrays on a glass slide. And you can purchase these again, each array, or we can do a custom service for you in which we would take patient samples and incubate the arrays with a patient sample. And then these are probed with secondary antibodies that are labeled with a fluorophore. And a common example is we would use 2 fluorophores, such as Cy3 for using different immunoglobulin subtypes. We could use Cy3 for IgA and say Cy5 for IgG. And then you can read the same array for 2 different signals. And then you can quantify the amount of signal on the array and see if a patient has antibodies to any of these viral proteins. So what are applications in general for antigen microarrays? Well, there are a number of them. So some of them are autoimmune disease profiling and diagnostics. And this is general. This is not specific for COVID. Cancer biomarker profiling, infectious disease or pathogen marker profiling, like we're talking about today. Allergen profiling, transplant evaluation, evaluation of the efficacy, toxicity, and other side effects from clinical applications of drugs and therapies, evaluation of human body responses to pollutants and impacts on health, and vaccine efficacy evaluation. Again, we also offer custom antigen microarray services, such as custom printing and spotting. So if you're not just limited to our pre-designed protein microarrays, but you can have us design a custom array for you, built to your specifications. Also, we have sample processing services where you can send blood, plasma, tissue, or other biological samples, and we will prepare them for processing and incubate with any of our pre-designed antigen microarrays or custom-built arrays. We also provide data analysis. So once the samples are processed and incubated with an antigen microarray, we will analyze the raw data, and the standard analysis service includes an Excel file of the net signal intensity for each antigen on the array, normalized to internal controls, and a heat map. And we have additional services available, including proteomic analysis, pathway analysis, and more. This is an example of the standard antigen microarray service analysis, where we provide normalized data in the term net signal intensities, or NSI. So we have the list of antigens on each array, and then these columns represent different patient samples, and then we provide the normalized signal intensities. We also provide a heat map where the signals are represented qualitatively by their intensity. So green is usually a low signal and a heat map, whereas red usually indicates a very high or significant signal. So we provide that in the form of an antibody score. So the higher the antibody score, the more significant the interaction between antibody and antigen. We can also provide additional analyses such as gene ontology analysis, or GO, which classifies genes and proteins based on known biological functions, as well as pathway analysis, which groups antibody-antigen interactions based on defined biological pathways. Okay, so now I want to move on to the next product line that's related to SARS-CoV-2. That is our BlazeTech Probe one-step SARS-CoV-2 testing kit. This is a qPCR-based diagnostic testing kit for active virus. So again, this is the schematic that I showed before of the SARS-CoV-2 genome. Many companies use primers that recognize the regions of the genome circled here in red, PP1AB, S, E, which is envelope, N for nucleocapsid, or NSP12. But we have designed this a step further to for our probes to recognize regions of the spike and the nucleocapsid genes that have been shown to undergo very few mutations. So the idea is that even when the virus mutates, all viruses mutate, of course, but in order to avoid false negatives, we've designed these probes against more highly conserved regions of the S and N genes, and that do not recognize other coronaviruses, neither the seasonal coronaviruses nor SARS nor MERS viruses. And so our BlazeTech Probe one-step SARS-CoV-2 testing kit has a number of high-quality features. First, it shows high sensitivity. It can detect as few as 10 copies of the viral target sequences. Second, it provides broad spectrum detection. So proprietary primer probe design enables detection of most SARS-CoV-2 strains. So again, this comes from designing the primers against more highly conserved regions of the S and N genes conserved among SARS-CoV-2 coronaviruses. Okay. It also provides high specificity. So we include UDG, the enzyme uracil deglycosylase, and DUTP in the reaction to reduce carryover contamination resulting in fewer false positives, and as well as high instrument compatibility. So the inclusion of optional ROX dye ensures compatibility with virtually all qPCR instruments in use. And so you can order these testing kits straight on our website, as shown here under ordering information. We have 4 different versions. These are based on, we have 2 different sizes as well as with and without ROX dye. And I must point out that the prices shown, even though we show prices here, these prices are only valid in the United States and Canada. If you live outside of North America or Mexico, you would need to contact your local distributor. Now, before I move on, I must note though that even though there's a high quality detection kit, the BlazeTech Probe SARS-CoV-2 detection kit is for research purposes only. It is not intended for diagnostic or treatment usage in humans. Now I want to move on to another product area, which is SARS-CoV-2 related plasmids. As I mentioned, GeneCopoeia is a company that started out providing genome-wide collections of human and mouse open reading frame or ORF clones. And so we have many clones that express ORFs for both the SARS-CoV-2 viral proteins as well as human host factors, as shown on our website. Okay, so one thing you can do on our website is you can order ORF clones in many mammalian expression vectors, over 250 vector types, that in either lentiviral backbones or non-viral backbones. And to do so, you would go to our website and you would just click on the product ID as shown here. And then it would take you to the page where you can select your different vector types. We also have several pre-made clones for various host factors, such as human, mouse, and rat ACE2. As shown on the website right here, we have a number of pre-made vectors that are ready to go. They're in stock, so all you would have to do is order them, and you would have them in either 1 or 2 days. Okay, so we have those for, again, for the human host factors such as human ACE2 and tmprss2, as well as other human host factors. Now, we also, I want to point out, have a large collection of genes in our library for other host factors, such as what was shown in a paper that came out in Nature a couple of months ago from Gordon et al., where they did a human interaction screen to look for other human proteins besides ACE2 and tmprss2 that interact with SARS-CoV-2 viral proteins. And what they did was they did an affinity purification mass spectrometry screen. So they engineered 27 viral bait proteins, and then they those were linked to a 2x strep moiety, which can pick up bait proteins that are then that they can pick up the bait. If they pick up the bait proteins, if they bind to the bait proteins, then the strep moiety can bind to mag strep beads, which they then elute, use biotin to elute, and then they identified by mass spec the various bait proteins. And when they did that, they found 332 interacting proteins, human proteins interact with viral proteins. Now, there's a number of caveats to keep in mind with such a screen. First of all, there could be possible false positives. This was not done on live cells. It's an artificial system, so they used cell extracts in this affinity purification scheme. So because it's an artificial system, it's possible that some, most, or even all the reported interactions are not biologically relevant. Likewise, it's also possible to get false negatives. So two of those proteins of note, ACE2 and tmprss2, were not identified in the screen. But tmprss2 is an enzyme, so it might not physically interact despite its essential role in viral pathogenesis. However, the paper went on to show that when they did a drug repurposing study to see if any of these drugs are, to see if any of these interacting proteins are targets of known drugs that are used for other diseases, and they found that 16 drugs that were previously approved by the FDA for other uses, as well as other drugs that are either investigational or preclinical, target these proteins. So, and then from literature searches, they found additional drugs that can target these proteins. So they found it, so in total, they were able to identify 16 drugs that target 66 of the 332 proteins. And so when they went further to show that some of those proteins, 16 of them have antiviral activity in vero cells. I don't want to go into too much detail about the paper, but the implications of this is that repurposing of existing or development of new antiviral drugs that act on host proteins instead of viral proteins can be a useful goal to combat the virus. And the reason for doing this is that drug resistance is less likely to be caused by genetic drift for host-directed drugs than for virus-infected drugs. And another implication is that this can be used for basic research to learn about virus life cycle and biology. So why focus on antiviral drugs? I mean, we're hearing every day about progress on vaccines, and of course, the ultimate way to eradicate a disease such as COVID-19 is using a fully effective vaccine. However, there's no guarantee that we will ever have or soon have a SARS-CoV-2 vaccine. And there are many examples in the literature, famous examples of the literature, of viruses for which a vaccine has never been developed, such as HIV, Epstein-Barr, or Rouse sarcoma virus. Sorry, not Rouse sarcoma virus, respiratory syncytial virus. A vaccine for Zika was developed in 2019, but has only been tested on macaques. In addition, there's never been a vaccine against any coronavirus shown to work in humans. So even though we've seen a lot of promising results lately, most of the vaccines that are in development do appear to be safe. They produce strong neutralizing antibody responses. However, we still don't know, we will still not know for at least a few weeks, if not months, if these vaccines are going to be effective. And so, because of that, it would be very useful to focus on antiviral drugs to at the very least, bridge the gap between having no treatments and between the time when we get a safe and effective vaccine. And in fact, it seems to be likely now that we will not have a mass-produced vaccine until at least early next year. So effective antiviral drugs such as remdesivir and others could fill that gap. So anyway, now I want to move on to, before I move on to that. So I mentioned that the Gordon et al. Paper showed that there are 332 interacting proteins, and you can order any of those, any of the genes encoding those proteins from GeneCopoeia in various vectors. Now I want to move on to other SARS-CoV-2 related products such as lentivirus and AAV. Lentivirus expressing SARS-CoV-2 viral proteins as well as human host proteins can also be ordered from our website. Lentivirus is great, as you might know, for studying gene function in cells, either in tissue culture or in animals. AAV, or adeno-associated virus, is more suitable for gene therapy style research or in vivo style research. And you can order lentivirus and AAV expressing any of the SARS-CoV-2 viral proteins or the human interacting proteins from our website. We also have CRISPR and shRNA solutions in case you want to do gene knockdown or knockout studies. And those can be ordered from the website. And we also have stable cell lines, such as cell lines expressing human ACE2 and tmprss2. So that's it. That's all I have to go over with you today regarding GeneCopoeia's SARS-CoV-2 viral proteins. I hope you found this presentation useful. So if you have any questions, I will go ahead and take questions now. I will start to read the questions. So how and when can we access a recording of this webinar? There will be a webinar, a recording of the webinar made available on YouTube in the next 24 hours. So you will receive an email if you registered for this webinar, and a link to the recording will be made available. Okay, so next question. Why do you only have pre-made lentivirus expressing luciferase and GFP? Do you have any other markers available? Yes, well, we don't have anything pre-made yet with expressing other markers. We decided to come out with virus expressing GFP and luciferase because of their utility. But of course, we can custom produce pseudovirus expressing virtually any marker you want or any other gene that you want. We would just need to look at the design and come up with a design plan. So, all right, does anybody else have any questions? All right, well, if not, again, I want to thank you all for attending. I really appreciate it, and have a good day. Take care.